Okay, my name is Mahvi Venkatesh. I am going to be a junior this coming academic year at UC Berkeley. I'm one of the undergrad researchers on the IGEM team.
Fantastic. So can you describe for me a day in the life of a synthetic biologist?
Okay, so as a synthetic biologist, you have to plan out your experiments, and a lot of times, like, you're just drawing pictures of plasmids on scratch paper and whatever you can find. It might be the whiteboard, or if there's no whiteboard, it might be the cover of the hood. So, I mean, you are trying to represent the things that are in all the little tubes that are sitting around in your freezer or on your bench. So that's like the design aspect of it, the part that requires a lot of thinking, understanding, kind of more of the knowledge aspect of it. And then there's actually the execution of it. It's probably what you spend a lot more time doing, because it's a lot of like manual pipetting work, kind of fun stuff. And also a big part of it is like keeping track of everything and where they are and what's in each tube, because otherwise you look at all these tubes and they all look the same. They're like, look at all your plasmids and they're all tubes of clear liquid, and if you don't remember which one's which, and if you haven't labeled it right, and like you don't, or even if you put numbers on them and you don't remember what part is what number and you didn't write it on a spreadsheet or on a paper somewhere, like, you have to start over. So a pretty big part of it is like keeping track of everything. And so I know, I have personal spreadsheets. We have spreadsheets for the team that we have as Google spreadsheets. So a lot, all of our parts have some sort of like ID number, whether it's a K number that's from the registry that was assigned to, like, we were assigned a certain number of K numbers for our team this year. So all our basic parts have those numbers, but then a lot of the other parts have some other shorthand identification that's usually person's initials and then their number. And so just like spreadsheets, keeping track of all of that so other people know what's going on, or so even you know what's going on with your own stuff, because honestly, MV5, I don't know what that is. I'd have to go look at a spreadsheet and figure that out.
Sorry, it's okay. There was an earthquake briefly. Knocked it. So how would you describe synthetic biology?
So synthetic biology, I think, to me at least, it's, you have these systems that already exist in nature, and you want to kind of break them down to their individual parts, and then you can reshuffle all these parts and put them together in lots of different combinations. And in the process of doing that, you can make a lot of different novel systems that do interesting things that you want them to do. So you can start with something that has one function and kind of take it apart, mix it around, and get something that does something different. So, I mean, I kind of think of it like Legos a lot of the time, because, I mean, these parts are pretty modular, so you can join them pretty easily and kind of mix and match them pretty well. And so, like, if you have this starting Lego structure and you kind of take it apart and you have all these Legos, you can kind of put them together to make something different.
And what is IGEM?
What is IGEM? So IGEM is stands for International Genetically Engineered Machine. And so basically there are these teams at different schools, and each team wants to engineer some sort of bacteria that does something interesting. And so, like, the teams get to pick what they want to do. There's no requirements about specifically what you want to make. You just have to like actually make something by putting some parts together. And so yeah, it's basically kind of synthetic. It's just synthetic biology, where students are coming together and working on these projects, and it's very student focused. Especially the way our IGEM team is run, we get like a lot of we got to have input in choosing the project, and in a lot of the design steps we've been working through with our instructors and our advisors, and just talking through it with them. Like, they may be giving us a lot of the information, but like it's really, we get a chance to understand why everything works the way it does, and they really encourage questions and things like that.
Yeah, so how did you come to be interested in synthetic biology?
Okay, so I first was introduced to synthetic biology when I was taking a class my first semester here. It was an introduction to biomedicine for engineers, I think was the title of the class. And so it was a survey course that covered a lot of different aspects of bioengineering, and among them was like one of the things we covered was synthetic biology. And I was really interested in this idea of kind of manipulating existing systems to like make new things. And I tend to like things that are small. I mean, even in high school I liked molecular biology and things that I couldn't see, that I could imagine, and I could see pictures, pictorial representations. But so I tend to like those things as opposed to the things that I can see, because those seem too obvious and intuitive to me for some reason. So like I really liked this idea of being able to manipulate things at the molecular level. And one of the projects that I heard about during that class was Chris Anderson's tumor killing bacteria project, and that was kind of like one of those things that I was like, whoa, this is so cool, I really want to do this. And so like that's how I first heard about it, and I kind of followed up with Chris after I heard his talk on that, and he was very receptive to the idea of having another undergrad work with him. And so I started working with him, and then it's all history from there.
So how would you compare those experiences working with Chris as an undergrad and the IGEM?
Okay, so working with Chris as an undergrad, a lot of what I was doing with him was kind of assays with mammalian cells. So I wasn't doing any design, pretty much. I was doing a lot more like execution type of things and assays, so like actually experiments where you get results. Whereas a lot of the things that I've done thus far with IGEM are like cloning and making parts. And so, I mean, so I was doing a lot less experimental design, so it wasn't probably as intellectually stimulating, I would say. But there were definitely like lots of things to think about. And because I was working on the tumor killing bacteria